examination of the cognitive deficits produced by subchronic ketamine exposure in rats by Chris Wood, Mohammed Shuaib, Institute of Neuroscience, Newcastle, UK. Chronic NMDA receptor suppression by NMDA receptor antagonists, i.e. ketamine, induce structural changes in neocortical and limbic brain regions, resulting in similar cognitive deficits to that observed clinically in schizophrenia. The objectives of this study. This study was designed to evaluate cognitive deficits following ketamine administration in rodents, assessed through an attentional set-shifting paradigm, thus allowing evaluation of cognitive flexibility in the form of discrimination acquisition, reversal learning, and intra-extra-dimensional shifts, i.e. the ability to switch between dimensions. This short presentation will cover key methodology and results from our study. Methods. 44 male Lister hooded rats underwent a 5-day subchronic treatment regime prior to habituation. There were 3 treatment groups. Vehicle, i.e. saline, ketamine at 10 and 30 mg per kilogram. Rats underwent a 2-day post-treatment cessation consisting of a simple odour and medium discrimination, followed by, within 24 hours, an attentional set-shifting task as shown by the apparatus above and the discriminations below. The order of the discriminations remained constant for each trial. The correct exemplars are shown in bold. The defined positions within the set-shifting apparatus A and B were determined by pseudo-random sequence. Results. Graph shows discrimination versus trials to criterion positive standard error. Subchronic ketamine treatment impaired reversal learning in a dose-dependent manner. Animals treated with ketamine 30 mg per kilogram resulted in significantly more trials required to achieve criterion for all 3 reversals, reversal 1, 2 and 3. Ketamine at 10 mg per kilogram only significantly impaired acquisition during reversal 2. Graph shows discrimination versus trials criterion plus standard error. Ketamine at 30 mg per kilogram treated rats made significantly more errors during reversals 1 to 3. Graph shows discrimination versus latency in minutes plus standard error. A clear trend emerged between ketamine exposure and latency. Rats treated with ketamine at 30 mg per kilogram required longer to complete RV1 to EDS. Conclusion. These results show subchronic ketamine treatment impairs reversal learning. Our findings support the impairments observed clinically following CANTAB assessment in schizophrenic patients. Hence, our data suggests subchronic ketamine treatment results in similar cognitive deficits. Thus, our results support the hypothesis that ketamine is an important pharmacological agent to model cognitive deficits in rodents.